Hello, and welcome to today's session. This session focuses on learning outcome 3, LO3. Discuss and evaluate the data collected and the conclusions drawn. In this video, we will explore how trends and anomalies influence our understanding of biological data. You will learn how recognizing patterns and identifying deviations can shape the interpretation of experimental results, helping to draw accurate and meaningful conclusions in biological research. Recognizing overall patterns is crucial in data analysis. Consistent trends indicate reliable data. For instance, in osmosis experiments, increased squash concentration causes potato chips to lose mass. This pattern holds true across various concentrations. Identifying such trends helps in drawing meaningful conclusions. Anomalous results deviate from expected patterns. In enzyme concentration experiments, one trial showed a longer reaction time. Such anomalies require careful evaluation. They might arise from experimental errors, like delayed timing or inconsistent mixing. Understanding anomalies aids in refining data accuracy. Accuracy in data collection is vital for reliable results. Using precise measuring equipment enhances accuracy. Consistency in measurement methods reduces errors. Human error can still affect data quality. Overall, maintaining accuracy ensures trustworthy conclusions. Evaluating anomalies involves assessing their impact on results. Anomalies like unexpected mass gain in osmosis experiments must be explained. Factors like chip thickness or surface liquid can cause deviations. Understanding these helps in minimizing their effect. Proper evaluation strengthens data reliability. The suitability of a practical procedure affects data validity. A well-designed method aligns with the experiment's aim. Control of variables is essential for consistent results. Limitations in the procedure can impact data quality. Evaluating suitability ensures reliable experimental outcomes. Suggesting modifications enhances data quality in experiments. Using identical samples reduces variation in measurements. Blotting chips uniformly minimizes surface liquid effects. Increasing repeat trials improves confidence in trends. Modifications lead to more accurate and reliable data. In osmosis experiments, squash concentration impacts mass change. Higher concentrations lead to greater mass loss in potato chips. This consistent trend supports the hypothesis. Anomalies like mass gain require careful consideration. Understanding these patterns aids in drawing valid conclusions. Increasing enzyme concentration shortens reaction times. This trend is consistent across trials, indicating reliability. Anomalies like longer reaction times need evaluation. Possible causes include timing errors or mixing inconsistencies. Recognizing these trends helps in understanding enzyme activity. Several factors influence data accuracy in experiments. Precision of equipment plays a key role. Consistent methods reduce measurement errors. Human factors like reaction time can introduce inaccuracies. Addressing these factors enhances data reliability. Anomalous results require careful analysis to understand their impact. In osmosis experiments, deviations from trends need explanation. Possible reasons include sample thickness or surface liquid. Evaluating these anomalies helps in refining data accuracy. Addressing them strengthens the overall conclusions. Identifying limitations in procedures is crucial for data quality. Manual methods can introduce errors in timing and measurement. Ensuring consistency in variables is essential. Recognizing limitations helps in improving procedures. This leads to more reliable experimental outcomes. Enhancing reliability involves addressing procedure limitations. Using automated methods reduces human error. Controlling environmental factors ensures consistent results. Increasing trial repetitions improves confidence in data. These enhancements lead to more reliable conclusions. Modifications improve data quality in osmosis experiments. Using a cork borer ensures uniform sample size. Blotting chips consistently reduces surface liquid effects. Increasing repeats strengthens trend confidence. These changes lead to more accurate and reliable results. Improvements enhance enzyme experiment accuracy. Using a gas syringe allows precise oxygen measurement. Maintaining constant temperature improves consistency. Automated timing reduces human error. These modifications lead to more reliable data and conclusions. Evaluating data trends involves recognizing patterns and anomalies. Consistent trends indicate reliable data. Anomalies require careful analysis to understand their impact. Addressing anomalies enhances data accuracy. This evaluation strengthens the overall conclusions drawn from experiments. Conclusion: understanding data trends and recognizing anomalies is crucial in biological experiments. Identifying patterns and addressing deviations improves the accuracy of results, while evaluating procedures and recommending improvements enhances the overall reliability of the data. Need help or have questions? WhatsApp us at 0748-4361-688 and our team will be happy to assist. Thanks for joining us today, and see you soon!